So let's have a look at pens. Everyday objects. What do we do with pens? Shove them in our mouths a lot. Lots and lots of people. We pass them from one person to another. Everyone borrows each other's pen. So do we clean pens? Can't remember the last time I cleaned a pen. So let's just have a look. Let's just see how many organisms are present on this pen. Bearing in mind that we're using the ATP monitor. So we're not actually counting organisms, but we're counting relative light units. And this is equivalent to ATP, which is an intracellular enzyme present in all living cells. If I was to send this swab to the laboratory to have the organisms cultured and grown, it would take at least 24 or 48 hours. But using this technique where we're looking at relative light units, which is equivalent to ATP enzyme, which we find inside cells, we can equate the numbers in general terms to microorganisms. On this occasion, there are 5,682 organisms present on this pen. That's very heavily contaminated. And just imagine if you're putting that in your mouth. It's nearly 1 × 10 to the 4, it's nearly 10,000. Oh, lots and lots of bugs. 5,682. So what I will do is I'm going to clean this now. I'm going to spray Steril-eeze on it. I want to give it a good clean to reduce any grime. And then I'm going to re-spray it with Steril-eeze. I'm going to leave this at least 30 seconds or 5 minutes. So I'll be back soon. So let's see what effect the Steril-eeze has had on the pen. If you remember, we had over 5,000 organisms on this pen. The one I frequently touch, pass on to friends and family, and usually sometimes ends up in my mouth. Look at that, none. That has been so effective on that pen. That's amazing. That is 99.99% reduction. This is the level of reduction that is claimed in the laboratory when they do the surface test. So again, it's proved that it's effective here.